皆様、本日はランチョンセミナーの中から日立ハイテク協賛 OhmX プラットフォーム for Human Whole Genome Structural Variation Analysis OhmX プラットフォームによる全ゲノム構造多型解析をご聴講くださり誠にありがとうございます。私は司会の日立ハイテク山本と申します。よろしくお願いいたします。本講演では2名のプレゼンターをお呼びし、Nabsys社による OhmX プラットフォームの説明、またユーザーからですね、実際の解析例を用いた解析のご紹介をさせていただければと思っております。1人目は OhmX を開発製造しておりますNabsys社のプリンシパルアプリケーションサイエンティストのジョン・トンプソン博士、もう1人は OhmX を使用した解析例をご紹介いただくテルアビブ大学のユバル・エベンスタイン教授です。本日はジョン博士からAdvances in Electro Genome Mapping と題し、エレクトロゲノムマッピング装置である OhmX の進歩について、解析ソフトであるヒューマンクロモソームエクスプローラー HCE との組み合わせにより解析された結果を技術面からご紹介させていただきます。ジョン博士はNabsys社のプリンシパルアプリケーションサイエンティストです。2011年から2014年までNabsys社で技術開発に携わっておりました。2023年6月にプリンシパルアプリケーションサイエンティストとしてNabsys社に再入社しております。Nabsys社を離れていた期間は他のバイオ企業でシニア R&D として勤められておりました。イェール大学で学士、カリフォルニア大学バークレー校で科学の博士号を取得後、ブラウン大学で分子生物学の研究助教授を務めておりました。次にユバル教授からマッピング・ザ・ダーク・サイド・オブ・ザ・ゲノムと題し、他の技術との比較も含めてユーザー視点で OhmX プラットフォームについて実力値と期待いただく点などをご紹介いただく予定になっております。ユバル先生はケミストリー&バイオメディカルエンジニアリングの教授で、単一分子ゲノミクスを主に研究するテルアビブ大学のナノバイオフォトニクスラボを率いております。また同大学の起業家センターの創始者の一人でもあります。またですね、血液検査を行う企業ジャックスバイオのファウンダーと CSO でもあります。科学、物理学、生物学、バイオインフォマティクスを組み合わせてヒトゲノムを研究しております。ユバル先生はゲノムマップ解析の第一人者でもありまして、今回 OhmX プラットフォームを使用した解析を実施していただいております。So, please join me in welcoming John and Yuval.
Okay, thank you, Tomo. I'm afraid I will have to switch to English instead of Japanese, and I hope that Tomo said nice things about me. But I'm pleased to be here in Sapporo. It's a very beautiful city, and I've certainly enjoyed my time here. And like to share some of the things that we're doing at Nabsys and talk about the development of the instrument that we have, that we've been working in very close collaboration with Hitachi. First, conflict of interest disclosure. I'm an employee and a stockholder in Nabsys, and so I get paid by them, and they paid my way here. Dr. Ebenstein is at the University of Tel Aviv, and he has no financial relationships to disclose. So he's an unbiased observer, or at least unbiased financially. You will find out that he has very strong opinions on many things. So what I'd like to talk about today is structural variation and its importance. It's really an understudied variant form because it's been very difficult to detect. And we're using the Nabsys system to do electronic genome mapping. And I'll talk about that as well as give an overview of the EGM detectors and the advantages, the analysis and applications. And I'll ask to hold questions until the end so that after Dr. Ebenstein talks, then we'll both answer questions in the time available after that. So this is what the instrument looks like. In fact, there's one here in the booth. To give you an idea of how big it is, it's not that large compared to many genomics instrumentations. And it really meets significant unmet needs with structural variation analytics. And it can either be used on its own, or it's also very complementary to short and long read sequencing. So structural variants, as I said, have not really been that well characterized, and now that people have been able to detect them more easily with long-read sequencing, the impact of them is becoming much better known. So that if you look at, let's see if I can make sure I don't blind anybody here. If you look at the germline variants, many of them are recurrent, all the ones in blue where they happen many times in individuals, and those structural variants are not ones you typically have to worry about. They may have some minor effects on phenotypes, but they're not pathogenic to the extent that some others can be. But there are some that are an issue where you just see them once or twice, and they can be cause of significant disease and phenotypic variation. With somatic variations, which occur frequently in cancer, but also in other situations as well, they tend to be unique. Even though there are some things like BCR-able translocations, in fact, there are thousands of different ones that cause that translocation. So these are not as simple as single nucleotide variants. And there are hundreds of thousands of them that have been found in human. They can be either inversions, insertions, deletions, duplications, translocations, just about anything you can imagine that DNA could possibly do, it has done. With the germline variants, they tend to be smaller. If you look at the average size, it's on the order of thousands of bases, whereas if you look at the ones that occur in somatic, they tend to be tens of thousands of bases. So they really can affect a huge amount of the genome. I'm not going to talk much about this, but the instrument can also be used for repeat expansions. Many of these repeat expansions are very GC-rich, very difficult to sequence, even with single molecule sequencing, because they are so extensive. With the electronic genome mapping, you're only looking at the length, so you don't have to sequence through these things, so that these are all the size that we can actually detect. And again, the critical thing with mapping as opposed to sequencing is we don't detect base-by-base variation. So the mapping gives you ideas about intervals of sequence, and with our tags, they're on average about 4,000 bases apart, so that you get that level of resolution. Some are just hundreds long, some are tens of thousands long, but you map it rather than sequence it. But as a result, you can see the lengths of these things fairly easily. So with next-gen sequencing, short reads are hundreds of bases long. Long reads typically 10 to 50 kb, although you can get some that are longer, so that that's the size range you typically see. If you're familiar with optical mapping, which is done by Genomics, it's similar in principle to what we're doing at Nabsys, but they use a labeling chemistry with optical tags, and they're looking at very long DNA, 150 to 500 kb. And the problem with that is that distinguishing tags is very difficult as they get very close together. The diffraction limit of light means that you can't get that great resolution. Instead, if you're using electronic genome mapping, we can actually go down to shorter lengths as well as the high end, but we're doing electronic mapping, and it's done using principles similar to what's used with Oxford nanopore sequencing, which is current blockade, where if you have an opening, in our case a nanochannel rather than a nanopore, and you have a voltage applied across that, you get a constant current. If you stick something in the way, like a double-stranded DNA molecule or a DNA molecule with a tag on it, you block that current. So you're measuring the voltage and the time between those voltage changes, so that you don't have the same diffraction limit of light. You don't have the expense of lasers to stimulate these. You don't have the expense of imaging systems to have to detect them. You're essentially just using lots of voltmeters and measuring the time between them. So in terms of the space, optical genome mapping does not really get into the range of short-read sequencing, whereas electronic genome mapping does. We can go up to the higher end as well as the shorter end. And the process is shown here. You have to isolate high molecular weight DNA, because if it's not long, you can't get long reads. We use a kit that's available commercially from New England Biolabs, a Monarch kit, so that's all in solution, done with glass beads. It's very straightforward and easy to do, takes less than an hour. The sample prep is also very straightforward, using a variety of nicking enzymes and polymerases, and it's all done in solution using just standard pipetters. And that can be, both the DNA isolation and sample prep can be done in less than a day, and then load the sample onto the instrument and let it run overnight, and the following day download the data and have it analyzed. and you detect the DNA. And there are two pipelines that we have, which are the discovery pipeline, where we do an alignment of all the different molecules and then map them against whatever reference you want. It's a de novo alignment, so you're not using anything as a basis to align these things. And you also have a verification where you're looking to see if a particular structural variant is there. If you have a hypothesis that you think something is there, you can look at it with the SV verification pathway. So I'm going to show you what this looks like. This is just a single nanochannel. You have two input pores where you have liquid being forced through by both pressure and voltage. Two output pores, then you have a nanochannel, which is where the detector forms. And although we don't use light in the running of the instrument, if you want to see the DNA, it has to be visible. So this DNA is labeled with light, and you can see it being forced by pressure and voltage through that nanochannel. And if you look carefully, what you can see is that the molecules speed up as they go through, and that's actually an important feature because it allows you to tell where you are in the molecule, and it allows you to deconvolute molecules that may be translocating together, because sometimes, as right there, you have more than one molecule going through at once. And so the speeding up is an important thing, and you can think of that as being analogous to a roller coaster. When you first start going over the top of the peak, you're not going that fast, but once the whole roller coaster is through, you speed up considerably. And the DNA, if you're holding up your arms, that's the tag. So the current version, generation of the nanochannel are 256 nanochannels in parallel, so it's the same sample run through all 256 at once. And this is what you get. Again, this is the detector here, and this is what the molecules look like. And so that you have a baseline voltage that's right here. The head of the molecule goes in, and then you get, this is the level 1 voltage where it's just a double-stranded DNA. And each of these spikes is a tag, and you can see that you measure the time between them. And this is a repetitive region of a centromere. It's a 200 kb molecule, and the distance between each of these is the same physically in the DNA, but what you can see is that the separation here in time is much more than down here at the other end of the molecule where it's going through much more quickly. So we have signal processing algorithms to convert these times into actual DNA distances. And again, in terms of the advantages, it's got higher resolution and lower cost and optical mapping. It also has reduced false positives. There's no amplification, there's no ligation, and you're looking at double-stranded DNA. So those are all things that minimize the number of false positives. And you can also easily distinguish molecules in the detector because of the large voltage changes when more than one goes through. And that's shown here where you have a trace where you have a co-translocating molecule here. And in fact, based on the width of these tags, you can tell which molecule they belong to, so you can actually map this entire molecule separately from this one and line them up with the position on the genome. So in terms of the two pathways, as I said, the variant discovery, it's what's there. Is it new or is it already known? Or the verification pathway is, let's take a possible variant and see if it's there. So we've been doing a lot of work with the genome in a bottle sample HG002. So no matter which pathway you want to follow, you do the measurements, and then with the discovery, you assemble the contigs. With the variant verification, you map them, and then map them to either the reference or the alternate. And then with the discovery, you compare it to a reference genome. With the verification, you just simply count how many things go to the reference and how many go to the alternate. And it gives you either genome-wide calls or verifies whether something is present or not. With the assembly, giving you the ability to look at novel structural variant discovery. It's more expensive and requires higher coverage than the verification, but the verification won't tell you something you didn't already know. And in fact, you can also verify the newly discovered SVs using the verification pathway if you like. So this is what the verification pathway looks like. This is actually a stretch of about 200,000 bases here. And each of these lines is a molecule with blue in one direction, black in the other. And these two red lines here are the boundaries of a 60 kilobase duplication. And you can see that over here, this is the same as 60 over here, and then it's repeated again over here. So many of these molecules are non-informative because anything crossing this boundary here, you can't tell whether it's in the reference or the alternate. What's important are the molecules that cross both boundaries so that the end of the duplication, the start of the next one is unique. And so if it's, you don't have that duplication, you wouldn't see that. So you can count just the informative molecules and determine that, in fact, that duplication is present. We've also been using the H3002 truth set, and I'm not going to go into details, but basically looking to see whether structural variants that are thought to be present are indeed present in the molecules, and the AUC is quite good. Where you start to fall apart is in the range of 300 to 500 bases, where it sometimes can be hard to tell whether the variant is there or not, and that's very dependent on the interval size. The longer the interval, the more challenging it is. So this is all going to be built into the confidence of what those are, but certainly the longer ones you have high confidence in when they're found. And the assembly-based analysis, the discovery is done using Human Chromosome Explorer, which has been developed by Hitachi. And this is output from there. In fact, this is the 60 kb duplication I mentioned. So each of these alignments here, oops, that's not what I wanted, shows you the where you expect to find a tag based on the reference sequence. And up here is where you're actually finding tags, and wherever you have the vertical lines, they match. And what you can see is the matching stops here and then picks up again over here. And so if you measure this, it's 60,150 bases there. And this is all left aligned here, so that these all match, and then the right thing is off. And this is the same thing with the right is lined correctly, and the left is off to the side there, diagonal. But this is the HCE program where you can actually do all these things, maneuver these, manipulate these. This is the coverage in that region. So there's lots of things you can do with the HCE to analyze your data. We've also been doing a lot with tumor normal cells. So this is from a paper that was published by NCI and collaborators. These are normal cells. There's essentially nothing going on in this region of chromosome 11. If you look at the tumor cells, you see an 82,000 base pair deletion, and it's, again, quite diagnostic. We have another one here where in the normal cells you see a short deletion, 475 bases in this region. You look at the tumor cells, you see the same 475 base pair deletion, but now you also see a deletion about 3,400 bases. This particular one was not picked up by BioNano when they looked at it. This is, these particular cells have been looked at with several different genomic techniques, Illumina, Oxford, Nanopore, PacBio, 10X, microarrays. So in terms of applications, I mentioned the rare disease and cancer. We're also able to look at cell and gene therapy, pharmacogenomics, and then as this technology gets out there in people's hands, I'm sure there'll be a lot of new things that people will be able to do. It's an integrated system where we supply all the reagents. And with that, I will turn it over to Dr. Ebenstein, who's going to talk about his experience with the instrument as well as lots of other things.
Hello, everyone. I'm very happy to be here. I don't like standing behind the podium, so I'm going to go over here. Before I start, I'd like to thank all my students who are doing all this work that I'll be talking about. And as you can see, I am talking about the dark side of the genome, and I want to tell you exactly what it is. But let's start with what are the questions that are interesting for me in science. So what I want to understand is what are the molecular determinants that make a cell what it is, define its properties. Now, in molecular biology and genomics, we mostly look at average population properties. So when we do proteomics or we do gene expression, we usually take the whole sample, we extract it from a mixture of cells or of tissue, and we get a profile. And when we get this profile of the population, we never know if this is really something that is distinct of all the cells, or we have some kind of mixtures of cells which give us, when convoluted, this average property. Now we are in a very exciting time where we can start to do single cell analysis, and Here is single-cell analysis of my blood. And you can see that if we do this, we see that every type of cell has a unique gene expression profile that allows us to unmix these properties. But how do these different cells get different expression profiles? They all have exactly the same genome. And this brings us back to the DNA level. To have gene expression, we need to have this information encoded in our genome. And when we think about that, and when I started this journey a little bit more than 10 years ago, we had two ways to analyze single molecules in genomics. First one, ancient but very, very powerful, cytogenomics and karyotyping. So here, each one is a single molecule. It's a huge molecule, but you can see the variation. And if you find out, for example, that you have 3 of these chromosome 21, this is the Down syndrome. Every information is here. You have to understand that even the tiniest feature is millions of bases because the chromosomes are packed. Now, on the other end of the spectrum, in the nucleotide domain, we have sequencing. Sequencing can give us mutations, but the problem here is that we don't have the context of the genome. It means that we can only see a very tiny fragment each time. There is a whole domain in genetics, which I call the gene domain, between 1,000 bases and 1 million bases, that is lacking tools for correct characterization, and this is the dark side of the genome. And this is because in this size range, we have some very interesting things that are happening. First of all, the genes, the length of our genes and gene complexes sit in this domain. And the other thing which is very important, the average distance between genes is about 100,000 bases. And a lot of interesting things happen in these regions. When we are talking about genetics, usually we are talking about only 2% of our genome, which means 98% of the genome is not included in this common genetics. And this is the dark side of the genome, which contains many, many secrets, and this is the side that I want to explore. Now, in order to do that, you need to use long DNA molecules. And one of the tools that we use extensively in the lab is nanopore sequencing. You may have heard of it. DNA is translocated through a nanopore. My video is not showing, but it's okay. But this is an example where you see a normal adjacent tissue from kidney, and this is from a tumor. And you can see on the very high level that very strange things are happening. You can lose entire chromosome arms. You can gain entire chromosome arms and entire chromosomes. And it's a very powerful technique because you can go down to the single nucleotide level and detect all kinds of mutations. But it cannot encompass the full range that we are looking for. And therefore, we also work with what is called optical genome mapping, very similar to what Nabsys is doing, but using optical signals. And therefore, we need to have an optical microscope in order to see all these fluorescent tags along the DNA. Just as an illustration, working very similar to what we have seen before from John, we have a very long molecule. Again, this is from my blood, 600,000 bases. And when you take it and you can scan it through the reference of the genome, you can find out suddenly that some things do not align correctly. And this means that I am missing about 7,000 bases here. Similarly, a lot of our genome is repetitive and contains repetitive elements. They are very hard to characterize with sequencing. But if you have very long molecules, you can actually see these arrays. And again, I have about twice the arrays versus the reference genome. And these things are very, very personal. So I've been following these technologies from the very start. I was the first person to have a BioNano prototype, and I took it all the way to where it is today, following this technology very closely. And this is why I was actually very excited when I could get my hands on electric genome mapping from Nabsys. This is me holding the first instrument in my lab in Tel Aviv. And you can see how compact it is versus other technologies. And the reason I was excited is because I think the potential of electronic mapping is huge. The scalability and the ability to quite cost-effectively analyze at very high throughput using these electronic signals is very attractive. And I decided to purchase such an instrument and start working with it. So we started by a benchmarking experiment. This is a classic genomics model, this GM12878. It's a B lymphocyte cell line that is used extensively in genomics. And when we ran this in the instrument, first of all, you can see this is the histogram of molecule lengths. So we have an average about 100 kb. And we could get about 205x genome coverage from the detectors that we used in this experiment. And what this allows us to do, because this is a very well-characterized cell line, and we know exactly where the tags are supposed to be, we can compare what is the probability of detecting these tag distances by our method. And what you can see here, that the range between 300 and 600 base pairs covers this range of 60 to 95% detection efficiency, which is the effective resolution. And this is how a molecule looks when it translocates through the channels. Now, when we take an overlook about these cells, we can see this is a copy number variation, each chromosome. And we can see that we have indeed the 2 copies of a normal cell, which is what we expected. And we also get a list of deletions and insertions with different size ranges from the instrument. But the real challenge is to see much more complex structural variations, because these cannot be easily found by sequencing techniques. And I'm talking about inversions. So when a piece of a chromosome just flips over in its size, when you sequence it, it will look exactly normal. If you don't find the exact breakpoints, which are quite rare, it will be very difficult to find. And indeed, we see that we get a whole array of inversions detected by this, and most complex, translocations. Translocations is when one piece of the chromosome is attached to a piece of another chromosome. And then we have these chimera chromosomes that are also very hard to detect, because if you want to detect them by sequencing, you have to hit exactly this breakpoint. On the other hand, when you have very long molecules, it's not easy, but it's much easier to detect. And here we had the opportunity to work with this cell line. This is called the circus plot. Every slice is a chromosome. And we know, because we've characterized this cell line very, very extensively, both with bio-nano and nanopore sequencing, that there are these 2 translocations that connect chromosome 16 and 7 and chromosome 22 and 14. And we were very curious to see if we can indeed detect this with electronic genome mapping. We were happy to find that both of these were detected. And let's see how it looks just by focusing on this translocation. So again, this is from our bio-nano experience. This is the assembled contig, and you see that one half connects to one chromosome, the other half to other chromosome. And again, with HCE, we see a very similar phenomena where we have our contig, and it aligns to both chromosomes, and this way we know that we have a translocation. If we're talking about inversions, again, we have molecules that span the complete inversion. It's 105 kb. But because we have very long molecules, we can see this inversion very clearly, and we catch it with both methods very efficiently. Deletions. Deletions are interesting, and it's interesting to validate them. In this case, this is a deletion that comes across all cells. You can see it's a deletion that we could find with nanopore sequencing very clearly. This is the deletion. We could find it by Nabsys, but this is a deletion that is rather small. It's less than 2 kb and was not detected by the bio-nano system. More complex SVs are SVs that are allele specifics. many times you will find that your molecules cluster into two groups. One group can span the whole reference, while the other group of molecules is shorter. And when you take this breakpoint and you separate them, you can see that they actually align very well to one side of the chromosome, very well to another side, and there is a gap in the middle, and this is an allele-specific deletion. And indeed, we found quite a few of these, and you can see this one, it's a very large deletion, 32 kb, detected by all three methods. You can see with the ONT that we have half of the molecules spanning the full range, and then half of the molecules that have this deletion. And again, with HCE, this is the genome reference, and we see that we have part of our molecules on the lower side which align to all of the reference, and then part of the molecules have this gap, and they align up to this point, and then they start aligning to a further region. A very, very preliminary comparison of the methods based on very small experience. We've only had the instrument for a very short while, so this is still bound to exception, but I think that this is important because it has some important features that are interesting for the users. Okay? So beyond the resolution, which differs between the techniques, these are the specifications of the flow cells that you use. So if you want to work with the system, then what is very interesting for you is many times, how much is it going to cost me to do this analysis? And what we can see very clearly is that there is a huge difference in coverage between the methods. And this means that if you calculate the per genome cost, then you find the real differences. And what we find is that with BioNano, if you take a special deal and you commit to doing 240 samples per year, you can actually get very attractive pricing. But if you are doing it like me and working quite sparsely, it will cost you about $1,200 to have a single sample run, and which will come to around $7 per genome. Oxford Nanopore, you have base pair resolution, but you have to pay for it, and the costs are quite high. Again, this is if you commit to buying at once almost 100 flow cells, then you can get down to $11 per genome. But if you buy a single one, it's cost you about $65 per genome. And with Nabsys, you have a guarantee of 100x per flow cell, which brings you to about $4.50 per genome. So looking forward, this is also a very scalable solid-state technology. So I can envision how the throughput increases dramatically with the same price, and you will end up with a much lower cost per genome. But genetics is not everything, and I'm actually especially interested in what is going on beyond genetics. And the puzzle is, how come that every human, each one of us, has its unique genetic code, and its unique genome is in all the cells of our body exactly the same code? Nevertheless, we have many different cell types that look differently, they act differently, and they still have the same genome. And this is because we have another layer of information, an epigenetic layer, which is encoded in chemical modifications to the bases. Most known is DNA methylation. This happens on cytosine, on C, when it comes just in front of G. This is why it's called a CpG. And these sites are scattered along the genome, but they tend to cluster around regulatory elements such as promoters. Now, when these islands get methylated, usually it means that the gene is shut off, and this is mainly because of a physical chemistry reason. These are very hydrophobic residues. It's like oil, and it's like you're putting a patch of oil on your DNA, and all the protein machinery cannot interact with it. Not very long ago, another epigenetic mark was discovered. It's an oxidized form of methyl, 5-hydroxymethylcytosine. And this actually triggers going back to cytosine, erasing this methylation and opening up the gene. So what we have here is a molecular memory, which is very, very important. At very early stages of development, epigenetics methylation is erased, and then each cell will develop its own unique methylation profile, which will dictate the destiny of this cell and what it will become, and we know for a while that each type of cell has a unique methylation profile. But not only in early development, this happens throughout life, and we know from identical twin studies that when identical twins are born, they have a very similar methylation profile, but as life goes on, even with twins that are both astronauts and lived almost the same kind of lifestyle, they have different methylation profiles because they did not live exactly the same life. This means that we have a molecular recording of our life imprinted in our DNA in the form of epigenetics, and it tracks down all of our exposures to chemicals, to food, psychological effects. All of this is basically encoded in our genome in the form of epigenetics. And my lab has been working for many years to develop chemistries that will allow us to attach tags to these epigenetic marks. In this case, I'm using fluorescence just to demonstrate. So we have these epigenetic marks on this piece of DNA, and we can light them up using our chemistries. And when you do that, you see immediately very striking things. First of all, different tissues will have different amounts of epigenetic marks. So even without genetics, you can differentiate them very clearly. Another example is in cancer. Cancer involves a lot of epigenetic changes, and therefore, these are samples taken from colonoscopy samples. This is a healthy colon, and this is a colorectal tumor. You see the difference in the yellow dots along this DNA. But why do you need epigenetic long reads? The reason is that between our genes, we have regulatory elements that can activate our genes from a distance. Remember that our genome is a bowl of noodles. All the chromosomes are entangled inside the nucleus, and they can interact one with another, and DNA can loop over and interact with very distant elements, and these elements are usually governed by methylation, and there are many, many examples, but this is one example of the ATL2 gene, where you see from GWAS studies that while it's distributed in the normal population, in the cancerous population, this enhancer is fully methylated. And we want to use these features and actually develop the ability to do multi-omic analysis with electronic genome mapping. What we intend to do is to use these chemistries to attach bulky tags on the DNA. And so then our DNA molecules will be labeled with genetic tags to tell us where we are in the genome, and with epigenetic tags that will give us this additional information. And then what we can do is we can use the amplitude of these electric peaks to differentiate between genetic and epigenetic information, allowing us to perform such multiplex analysis. And my vision and my plan to work with this system, and the reason that I am involved with it, is the ability to really do multi-omic electronic mapping where we have all the structural variants and the structure, genetic structure of the genome, and overlaid we can have these epigenetic tracks that will tell us both if the tissue is healthy or sick, and also what is the exact epigenetic profile of these certain modifications. With this, I'd like to thank you and thank all the people that gave me money for this research. This is our lovely campus in Tel Aviv, and hopefully we will have more peaceful times, and you can all come visit us in our beautiful country. Thank you very much, everyone.
Thank you both for such inspiring talk.
And so we'll take questions now, but there's one thing I'd like to point out. We share Yuval's vision of what we'll be doing with epigenetics, but right now that is not a protocol that is available, but hopefully he will get that developed so that we will be able to do that in the near future, but it's not available quite yet. Questions?
ありがとうございます。ここで質問を受け付けたいと思います。ご質問ある方はお手を挙げていただければと思います。I have a question for Yuval. So, is that okay to be? Yeah. I know you're analyzing for many kind of diseases, but first, after you got the reagent and detector from Nabsys. Which kind of diseases you would like to analyze first?
Okay, so we mainly focus on cancer because, as you maybe, if you remember that I've showed it, cancer genomes tend to be structurally aberrated. A lot of very complex things happened. This is mainly because of DNA damage and repair with non-homologous recombination that gives all kinds of things. But it's also very important for neurodegenerative disease and for muscular dystrophies, because these also tend to be very structural variant sensitive.
Thank you very much.
Okay, this is probably not related to what you just said, but I would also like to point out, I mean, you'd asked about the diseases that these technology can be used for, but in fact, it's not simply disease study that we're looking at. So that, one of the products of diagnosing all these rare diseases is you're now going to be able to possibly do cell or gene therapy with these patients. But as you do, generate these cells or gene therapies, you have to worry about, well, what is the impact on all the other cells? What is the impact on cell therapy with genomes that could be altered? So that this technology can also be used to characterize those vectors, their impact on the genome, look at the cell therapies. Do the cells have the genome they're supposed to have, or have you modified things? Have the gene therapy vectors integrated someplace you don't want them to be? So these are all things that can be done. So it's our current focus is on disease diagnosis, but ultimately it will also be on characterizing things that you're doing to cure those diseases, because both gene therapy and cell therapy, they're still, for the most part, experimental, but as we do more and more of that, there'll be more and more need to characterize them, because it's not just rare diseases. And certainly there have been a number of deaths related to gene therapy, and it's important to understand why that happens. Is it because of the DNA vector? Is it because of some modifications to the genome? Or is it completely unrelated? So those are also the kinds of things we're looking at. And that's why we're working with people like Yuval to try and understand, well, what are the other applications that can be done? And I'm sure that people out in the audience have thoughts about, well, gee, maybe you could do this, maybe you could do that. And there are lots of things. And again, this is really, it's not just detecting DNA tags, it's detecting differences in shape. So although we're focusing on double-stranded DNA at the moment, you can also envision other things going through those nanochannels and potentially detecting different conformations. And so that the possibilities are virtually endless in terms of what is it that you can see with this technology. Now, we are a very small company, so most of these things we're not working on. Hitachi is a very large company, and we're working with them to help try and bring some of these things to fruition. But hopefully over time that will happen, and we're thankful that they're working with us so that we can bring this technology out and expand it into areas that your imagination will hopefully think about.
この技術は新しい開発のところまで今お話をいただいたんですけども、どんどんいろんな解析ができるようにしていきたいと思っておりますので、ぜひよろしくお願いいたします。皆様の中で疾患はあるけども原因がわからないですとか、大きな構造多形を見てみたいというお話があれば、ぜひお声掛けいただければと思っております。お二人に拍手いただければと思います。本日はお忙しい中、ランチョンセミナーにご参加いただきましてありがとうございました。